Hi everyone, this is Mr. Hall and this is a quick video about enzymes and required practical that you need to know for the AQA GCSE. This is more to do with the sort of data analysis side of things. I talked earlier in the previous video about the planning side, so hopefully that's been helpful, but today we'll talk more about the sort of how you deal with data and exam style questions that you might get based on that kind of thing. So the objective of the video as follows: we're going to describe the effect of pH and temperature and enzyme activity, although I'll probably do another video which goes more into the factors that affect enzyme activity later. We'll interpret data from tables and graphs and also describe and explain trends in data from enzyme practicals. So first off, what you'll typically get if it's an exam style question is something like this, where you'll have to basically make some inferences and not predictions, but certainly conclusions based on data which you might obtain from an experiment. And it'll be something like this. So from this, what you can see is that you've got the pH, and then what I've got here is a time until the enzyme stopped working in minutes. And then when you're plotting this data, you may be asked to put this on axes. So I've got the pH at the bottom here, and I would probably number it from maybe 0, 2, 4, 6, 8, and 10. And then over here, this would be time. So I'll write time over here, and that's obviously in seconds. And then it goes up to 5, so just make it easy, we've got 0, 1, 2, 3, 4, and then 5. Now, if I plot this data, so at pH 4, it's got time of 0, so I put a cross there. At pH 5, it's got a time of 1, which is there. pH 6, it's at 4. And then pH 7, it's got a pH of 5. And then it goes back down to 4 again at a pH of 8, and then it goes down to 2, which is quite low, it's over here, and the pH of 0 at 10. So that's kind of the graph you've got. And then if you wanted to kind of match this up with line of best fit, you can use a curve for this, so it would look a little bit like that. I haven't done it as straight as I potentially could, but it would basically look like that. Now, there's a few inferences you can make from this. First off, you can see that anything from pH 4 and below, there's no activity here, all right? So basically that tells you that the enzyme is not working. There's also no activity here. All right, so because there was no activity means the enzyme does not work at these pHs, okay? So enzyme does not work. Okay, now here, this shows me that there was 5 whole minutes until the enzyme stopped working. That's our peak. So this tells us that this is the optimum pH for this enzyme to work. And there's lots of different enzymes which work at different pHs, but we know that for this enzyme, it works perfectly at pH of 7, okay? So basically, that's how you analyze any data that you get on these kind of things. And you can make lots of conclusions based on that. Now, in terms of exam style questions based on analyzing enzyme experimental data, it can go in a number of directions. One is just to identify the enzyme from a graph. Now, if you remember the graph I just showed you, we had the pH of like 4, 5, well, let's do, sorry, 2, 4, 6, 8, and 10. And for our graph, it basically showed a pattern. I might extend it just to make it a bit easier for you to understand, but it basically showed it going like this. All right, and you can see here that the optimum pH is a pH of 7. Now, because that's the case, it's a pH of 7, which means it doesn't work in the stomach, which is acidic. It doesn't work in the small intestine, which is alkaline. So it can't be protease. It can't be lipase. It has to be carbohydrates. All right, and carbohydrates, such as amylase, you'll find in lots of places in the body, but in particularly in the mouth, made by the salivary glands. Now, if you had a pattern that looked a little bit like this, where its optimum is at a much higher pH, let's say like that, because the optimum pH is much higher, around sort of 1 or 2, that would tell you that it works best in acidic conditions, and that means you'd probably find it in the stomach. And because it's most effective in the stomach, it would have to be protease. Likewise, you might find an enzyme that works best in alkaline conditions, so it might have a profile that looks a bit more like this. Now, if that's the case, this one has got its optimum pH is around sort of 11. And that would tell me that it likes to work in alkaline conditions. So it has to be working in the small intestine. And as a result, it's likely to be a lipase. All right. So you might get a graph with these 3 different types of peaks, and you have to be just quite careful about identifying which enzyme matches with which curve. So that's something you might get. Now, you might get it as a graph to identify. You might get data instead. So you just have to be quite aware of that. Now, when you are plotting data, and you're often asked to do this, and it's worth loads of marks, just be aware that you're only going to be given half a block of tolerance, okay? And I'll explain to you what I mean by that. So again, we've got pH at the bottom here, so let's go for 2, 4, 6, 8, 10, and then 12, and this is obviously pH. And then we've just got enzyme activity here. I can't write this particularly gracefully, but I'm just going to write enzyme activity, and it's in arbitrary units. Now, sometimes in these questions, they might not use enzyme activity in arbitrary units. They might have, similar to the one I showed before, you know, time before the enzyme stops working, or like the size of egg white getting smaller because of protease. They'll use loads of weird examples to try and confuse you, but basically it's just the rate at which either the enzyme works or doesn't work, or the rate at which something gets digested, which is obviously going to be another video as well. But just going back to this, my maximum number is 29. So let's go up in, let's go 4. So 4, 8, 12, 16, 20, 24, 28, and then 32. Lovely, works perfectly. So when you're plotting this, if we look at my first data point, pH of 2 is got an activity of 4. So that would be bang on here. Now you have a half block of tolerance. So what that basically means is if I plot it there, or there, right, either of those points, I will get away with it. But if I plot it 1 block above or 1 block below, I lose the mark. So make sure you plot really carefully. One of the best bits of advice I can give you is when you plot this, use crosses, don't use dots, because dots can be quite inaccurate, hard to read for the examiner, easy to lose a mark. So you can see that pH of 3 is at 7, which is around, so 6 is in the middle there, so 7's around there. pH 4 is around 11, which is around here. pH 5 is 14, which is bang in the middle there, and so on. So you can see that slowly but surely you can build up your data count like this. Now, usually this is worth 4 marks, okay? So you get 4 marks for plotting a data and putting the line of best fit in, which I think is an absolute bucket of marks, really. You also get a mark for your line of best fit. There's loads of marks basically available for these kind of questions. So spending a bit of time getting it right really does pay off. So my data at the moment looks a little bit like this. I think I'm doing it reasonably well. There's 27, pH of 11 is 24, and then pH of 12 kind of descends to 17. So that's basically my data set. So your line of best fit, now it doesn't have to start at the origin, okay? But roughly what you've got here is it goes up and then goes down, and that's okay for line of best fit. And then you know obviously you do a much smoother curve than what I've done here. It doesn't have to go through all the points, it just has to roughly go through the points, and that's how you would do it. So just make sure you do your line of best fit like that. And then sometimes might be asked to identify the optimum pH. Optimum pH, as I mentioned before, is basically the maximum pH at which it works. In this case, it's around a pH of 9, all right? And you can see that from the data as well, so you can identify it from both. Okay, couple other things. Sometimes we asked to make conclusions from data. Now that may be conclusions from a graph or conclusions from a table. Much easier from a table in my opinion, but from a graph you can still do this. It's quite straightforward how you do it. So first off, you have to do a really detailed description of the trend. So if you look at this bit of data here, you can see that as I go from pH 2 to pH 9, the activity goes up, all right? So you can see it increases from there to there. So, and that would be your first statement, increase in activity from pH 2 to pH 9. And then you can see it goes from pH 9 to pH 12, it goes down, and that would be your second statement. So just make sure you mention the pHs at which you see increases and decreases, and mentioning the actual units or the values is useful as well. So you could say increase in activity from pH 2 to pH 9, but then include the numbers 4, through to 29, and then here 29 through to 17. That's quite good to do when you're writing conclusions. Obviously, we've mentioned optimum pH, so I don't need to mention that again. The other thing as well to mention with your conclusions is where the enzyme will be found. And I've touched on this already, but you can see that the optimum pH is alkaline. As a result, you're most likely to find this in a part of the digestive system that has an alkaline condition, and in this case, it would be the small intestine. That's asked quite a few times, so just be aware that you may be asked that. And you might also be asked for the name of that. In the small intestine, you know it's going to be an alkaline condition, so it'd have to be lipase. So remembering those basic facts is really important for these kind of questions. You might also be asked for suggestions to improve the experiment. Now, those suggestions can go basically in one of two ways. Now, one of them is greater precision of data. Now, breaking that down is smaller intervals. Now, you can see in this experiment, I've got pH of 4, 5, 6, 7, 8, 9, and 10. Now, the problem is that I've got two top values of 5, so I don't know whether it's pH 6 or pH 7 that is the optimum pH for this enzyme. So the best way of actually working that out is by doing smaller intervals of pH. So then what I could do is go, instead of going up in values of 1, I could go up in values of 0.5. So I'd also get a reading at 4.5, 5.5, 6.5, and so on. And that will tell me, give me a bit more information as to what the optimum pH of that enzyme is. I would also use more precise measuring equipment. So here at the moment it says just 5, but if I've actually got a much better way of measuring that, I might get that to like 1 decimal place or 3 significant figures. So that might show me that this is actually 0.12 and this is 2.36 and this is 5.18. This might be 5.37 and so on. And then that makes it much easier to identify what the optimum pH is. So those are two different ways of getting better, more precise data. The second thing that's actually quite effective as well is getting a repeat. Now, at the moment, I've only got one test of this, but if I was being smart, I would do a second test, because in my second test, I might find that it's 0, 0 here, and then I might get 2 again, and then I might get 4 here, and then 5, and then 4, and then 2, and then 1. And by doing that, I could work out an average from the repeat, so I could work out a mean, and that would give me much more reliable data. So that's just a summary of all the different questions that you might be asked on the analysis side of the Enzymes Required lab, and best of luck with your questions.